Thank you, the chair, and thank you for the organization of this session. It's very great. So today, I will talk about something different. So we do the simulation and the modeling. So for these slides, mainly simulation and modeling results. So I agree with Professor Chen that after the lunch, there's no equation, so there's a lot of pictures, a lot of summarization of the key results. So here some basic concept of for our lab, what we are doing in these civil and environmental departments. So traditional civil engineering studied the structure at microscopic level. But as the development of those nanotechnology, we start to observe that the structure at those microscopic level also play a very critical role to control the material property. And it interplays with these macroscopic structures to determine the entire performance of the structure. So for our lab, we start from the most fundamental level, the atomistic scale, and we scale up to the large scale. The reason we look at those biological material, look at this Ashby plots, Espinosa, oh yeah, Professor Espinosa here. So for engineering material, you have this banana curve, but for many biological material, it's the inverse banana curve. So it's a combination of toughness and strength, stiffness. Also, it have other property advantages for those biological material, like the self-healing, adaptability, and compatibility, and also those multifunctionality. So we want to study what's the mechanism inside those biological materials, and then we want to use those information to reverse engineer of those materials. And for example, we look at those three kinds of materials, the collagen and the cytoskeleton. This is intermediate filaments as example, and those beta sheet structure inside the spider silk materials. And they present the material inside the cell, outside the cell, but inside the tissue, and the material outside the tissue. And we do the comparative study. So by comparing the mechanical property and the structure of those different materials, we want to qualitatively obtain what's the mechanism that makes those materials have different, distinct mechanical properties. And we do the multi-scale modeling. So start from the fundamental level. So for organic materials, if the force field exists, I mean, for example, for protein, charm force field is very well recognized. So we just use that force field. If that some force field doesn't exist, so we do some DFT calculation to implement those interactions. And then we do the classic MD simulation, also some combination of MD and molecular Monte Carlo simulation, like replica exchange simulation. And we characterize its mechanical property, and we scale up to the continued level. So for example, here's the movie shows the stretching of the coil, and at the end of it breaks, start to separate apart. Those alpha helix under the stretching force turn into the beta sheet structure, and it's significantly increase the strength between before separation of those alpha helices. And the first part will be the molecular mechanics of intermediate filaments. So this is a kidney epithelial cell. So you can see those branching structure, those the keratin intermediate filament inside the cell is very random and branching. And the basic building block of those intermediate filaments is those dimers and tetramers. They are largely composed of those alpha helix structures. So in the simulation, we run the implicit solvent in CHARM and the explicit solvent in AMD that developed in UIUC. And we validate our result against the experimental measurements for those equilibrated structures. And then we perform the mechanical loading. We fix one end and applied force to the other end and see how the structure, the pooling structure changed during the pooling process. And we found that those structures transit from alpha helix to beta sheets, and we give a theoretical model to explain this process. Actually, that's the unfolding of those hydrogen bonds, and because of the hydrophobic effect, those strands squeeze together and they form the beta sheet structure. And those processes also found in experiments. So after stretching, those filaments become much thinner, and their secondary structure transits from alpha helix to beta sheets. And we compare the force extension curve that obtained from simulation against the AFM stretching curve, and we find qualitatively agreement, and each section corresponds to different mechanical molecular structure change. So for the first region is just the elastic deformation of alpha helix. Second region, unfolding of alpha helix, and this stiffening region, that's the structure. After structure transition, those alpha helix change into beta sheet and makes the force much larger. And we use those force extension curves to train our network model to simulate if there's any crack, how this network will behave. So it has the background of this nuclear laminar. So it's the inside the cell's nuclear membrane, and it makes the cell's nuclear have this mechanical property. And so we compare it against silicon. So for silicon, it's very brittle. So if you have a defect, you load it, and the crack will propagate. But for this network, if you have an initial crack inside and you deform it, this crack will significantly deformed before it goes to rupture. And also, if we change the initial length of the crack, we make it longer and longer, we still be able to stretch the entire network beyond 100% string. So, and it's because these force extension, this stiffening region that enable the material to diffuse the deformation energy inside the entire material, so become more difficult to rupture. And we also discussed the crosslink effect, and we compare two different cases, one with a strong crosslink, one is the weak crosslink. And we found that for a strong crosslink, the force extension curve, as shown in this red curve, is actually weaker than the weak crosslink. It's because for this network, if the crosslinks are weak, those crosslink will sacrifice itself before a rupture of those filaments. But if the crosslink is too strong, those filaments will break before a rupture of those crosslinks. And the second part I will introduce is our study about the characterization and the synthesis of the spider silk. So a spider silk is a natural material, so spider use it to catch the prey. And this very regular network actually are composed of the protein structure, very random protein structure. So some parts are beta sheet crystal structures, most of the part very disordered amorphous structure. And here we used a computational technique called replica exchange. It's the combination of Monte Carlo and molecular dynamics simulation. So its advantage is you take given the sequence, it enables to give you many possible conformations, but not only one equilibrium structure. And it rates each structure with a probability number. So you can use all those structures to do the statistic study. So we run that and obtain some possible conformation for the spider silk, and we stretch it. we find this force extension curve, as shown earlier, Dr. Chan's slides. Those force extension curve are observed. So you have this toe region and the stiffening region. And we also, in the simulation, we can see how the secondary structure change. So we can see that during the pooling process, the beta sheet content get increased and the amorphous region get decreased. And for the number of hydrogen bonds, we found first decrease and increase and drop. So the first decrease corresponds to the unfolding of those amorphous region. And this increase corresponds to the beta sheet formation. And this decrease because the beta sheet, the force is too large and the beta sheets start to slide against each other and those hydrogen bond breaks. And we can run simulation and systematically discuss the mechanical property of those amorphous region and this beta sheet crystal region. And we found for this beta sheet crystal region is very sensitive to its size. So if this size is small, so you pull it, the strand just slides against the other strands, and the force is high. But if you have a very long beta sheet crystal, its deformation will couple with the bending of this entire beta sheet crystal, and it actually become weaker during the pooling. And use those mechanical test results. We built bead spring network model. Works like a finite element model, and we use that to investigate how the size of those spider silk affects its flow tolerance. So if the silk is very thick and you have the crack, it will be very easy to propagate. The crack will make the fiber become two fibers. But if the fiber is thin, and this crack will be difficult to propagate, and the deformation energy will go through the entire rest of the material. And that explains why in spider silk usually that's very thin, because if it's thick, it will be easy to branch and become fuzzy, and it will be not able to support loading in one single direction. And that called back to this how spider produce those silks. So each silk runs several tens of nanometers to several hundreds of nanometers. They're very thin, and once they produce, they form the bundle, and that's one single silk. We can see it from the spider web. And we collaborate with Joyce Wang in Boston University to use microfluidic technique to fabricate those spider silks. So we use the silk peptide produced by David Kaplan in Tufts University, and use that technique. Those silk peptides can fabricate and get aligned to produce the man-made silk material. And we make some structure silk fibers like shown here. And in our simulation, we can systematically discuss, we build a DPD model, and we can study how the shear force induce forming of those silk fibers. And we can change the ratio between the hydrophobic region and the hydrophilic region. And here it shows if we have once our peptide have too many hydrophobic region, it will form those micelle structure instead of a very homogeneous structure. And we also we are also discussing the shear rate effect in forming those structures. And we do collaborating with Jennifer Lewis, that originally from UIUC, where she's now in Harvard University. We use 3D printing technique to make those artificial spider silk. So I hope those silks are made from the real silk, but actually right now it's just PDMS, you can see here. And we can characterize the size they made from those printers. And this PDMS actually, they are very elastic, and it's not quite sensitive to the loading history, at least several meter seconds, those kind of slow pooling rates. And we used a rubber model to model those PDMS material, and it gives, in our simulation, the force extension curve looks like this. And in the experiment, they only did initial stretching, so because limited by the boundary condition, so they didn't do they didn't stretch the fiber until its rupture. It also only goes here, but you can see a good agreement between our model and experimental measurements for those material. And then we can do the simulation to simulate how a prey gets catched by those spider web. So we do the simulation and also do the experiments. So the force extension curve for those structure under point loading have a pretty well agreement, especially for larger number of spiral silks. And for this three-ring experiment, it's not a good agreement because maybe there's some inhomogeneous once it gets printed out. Yeah, this web was the first to print. We need to go back and maybe do the improvement. And here shows some comparison between the experiments for those point loading and the simulation results. And then here switch to the third topic is about the interface. So we studied the mechanics of biological material, but it need to have the interface to make the material into an integrated system. So here we study one example that represents those interface, that's the muscle adhesion. So those muscles, so they found at seashore to attach to those stones, glass, and wood. Even they found they can attach to those Teflon surface in underwater and in salt condition. So it's very interesting to ask how they managed to attach, and the attachment is so strong. And this attachment is actually a multi-scale problem. So at large scale, you can see this, and for the microscopic scale, these each of those filaments, they have the gradients inside for their material composition. And at very interface is the attachment is exerted by those muscle foot protein, and for each muscle foot protein is rich of those DOPA structure, and this DOPA structure originally comes from the tyrosine, and under the certain chemical interaction environment, it becomes those dopamine for those adhesion. So we start by looking at how the DOPA attach to the silica surface, and here we use the charm force field, and we have the 0.5 mole per liter sodium chloride concentration inside solvent to mimic the sea water condition. And after equilibrium, we see that those single dopamine attach very flat on the top surface of the silica, and is qualitatively agree with the experimental observation. And also we measure the free energy landscape for those single dopamine to detach from this surface. And then we do the first derivative of the free energy landscape, and we got this force extension curve. So the peak force is around 743 piconewton in simulation, and comparing to the AFM test, it's pretty well have a pretty well agreement. And then we are confident about our method, and we do a larger scale study. So for this single alpha helix composed of this DOPA and comparing to this tyrosine, we can see for the tyrosine, this alpha helix mainly, I mean the mass center of this alpha helix still the axis of this helix structure. But once it changed to DOPA, you can see the mass center actually get closer to the surface of the silica. And we check our simulation and see that those DOPA side chains get attached, get closer attached to the silica substrate, and they form those double hydrogen bonds to the silica substrate, and that increase the adhesion between the alpha helix and the substrate, and also the hydrogen bond number. So for the tyrosine, it's very small, so it's 5 times smaller than this DOPA alpha helix. So that means this DOPA is not only adding one more side for forming the hydrogen bond, it also, the stability of those hydrogen bonds gets significantly increased once it forms those double structure. And then we scale up our study, so we want to study how the protein, the muscle food protein attached to the silica substrate. And we use the replica exchange to produce many possible conformations for those proteins. And then we study each of those conformations, what's their surface energy, to attach to those silica substrates. So for this MFP, muscle food protein 3, is 21 to 28 millijoule per meter square, and for this MFP5 is also a similar number, and it's similar to the gecko adhesions energy. And then we use those energy we measured from the atomistic simulation and combined with continuous models. So these models similar like the shear lag model. So the detachment force is caused by the stiffness of the material, and also the surface energy between the material and the substrate. And we use that combining with our measurements of the surface energy, we obtain the detachment force for those adhesion plaque is around 0.2 to 0.3 Newton. And we do the tensile test for each of those single fibers, and we observed that the average for those detachment force is also 0.4 plus minus 0.1 Newton. So the simulation results combined with theoretical predictions have a good agreement with the experimental results. So we're happy, and then we want to study how the entire muscle attached to those substrates, and we collect our samples in Boston Harbor area. We put our cage inside those islands, around the island, and we harvest those samples. So yeah, they attach to many different substrates, and then we do the tensile test to measure how the stretching force can be once we detach those muscles from the substrates. And the static strength, so once we use a very low pulling rate, 0.1 millimeter per second, and it's 5 to 6 Newton. But this force is very smaller than the mean value of the dynamic force generated from waves. So in literature, around that area is measured to be 17.8 Newton. So from that, we wondering, so maybe those muscle adhesion threads are more than simply affixing the muscle to the substrate. So for those so we discussed the composition of those muscle threads. So they are the combination of the soft material and the stiff material. So it's not a homogeneous material, but it's a heterogeneous. So and they have a distinct force extension curve for 2 different materials. And we model this bias thread network, and in our model, we can systematically change the ratio between this soft material and the stiff material. So here, some tests that for the single thread detaching, how our model comparing to the experimental results. So it's pretty well agreement. And then we, in our simulation, we give the initial velocities to those muscles to mimic the impact on those muscles, and we study how the force distributes inside the network. And it seems that for this stiff material, the loading is become lower than the softer material. So it means the softer material able to damp out the impact force once the force transduct from the muscle to those muscle threads. And once we systematically increase the impact velocity, and we find that critical impact force is 48.5 Newton, which is over 900 that of the static strength. If you recall that static stress is 5 to 6 Newton for those muscles adhesion. And we also, we systematically change the ratio between the soft and stiff material. And we find that at 20% of the soft material, that gives the minimal reaction force on this network. So, beyond, so other than this number, so if you have more softer material, the speed will be very high, and viscous force will dominate. Play a dominant role. But if you have more stiff material, the viscosity of the material will be dominating, and that also leads to the larger reaction force. So here it shows that this 20% of soft material and 80% of the stiff material makes the optimal case for those creatures to attach to the substrate. And here I just want to highlight that by starting those biological materials, we want to migrate our knowledge into the improvement of those engineering materials. For example, this is the recent work by Professor Buehler and Professor Espinosa on those carbon nanotube young study. So those structures, they have the stiff material and the soft material, stiff as the carbon nanotube, soft as those polymers. So this largely represent what we've seen in those protein materials. So like the beta sheet structure, backbone stiff, and the hydrogen bonds very weak. So some knowledge that we gain from those by studying those biological materials, we expect to apply that to studying those engineering materials and improve the performance of them. So I want to acknowledge those people in my group, and here's Marcus Buehler. He cannot make here, but there are a couple of people work on different projects. And I also want to acknowledge our collaborators, David Kaplan, Joyce Wang, Espinosa, Jennifer Lewis, Lauren Kaplik, and also the funding, the support from those fundings, Air Force, Navy, and the NIH. Thank you very much. And I'm pleased to take any questions you have.